Alan Gray here with the Microbiome First Summit, underwritten by the World Asthma Foundation. We're pleased to introduce our next speaker, Dr. Emmerin Mayer. Dr. Mayer is a gastroenterologist, neuroscientist, and distinguished research professor in the UCLA David Geffen School of Medicine and founding director of the UCLA Brain-Gut Microbiome Center. Dr. Mayer's research looked at alterations in the bidirectional communications within the brain-gut microbiome system and their role in chronic inflammatory and functional diseases of the gut, obesity, and cognitive decline. Today, Dr. Mayer presents his session, The Gut-Brain Axis and the Microbiome, Mechanisms and Clinical Implications. Dr. Mayer, over to you.
The topic of my presentation today is the gut-immune connection and specifically what role your microbes play in it. I would like to start my presentation with a disclosure of potential conflicts of interest and give you a brief overview of the topics that I will take you through in these next 45 minutes or so. How we ended up having two brains, microbe speak, development of brain-gut microbiome interactions, stress gut and the microbiome, putative brain-gut microbiome disorders, and challenges. I would like to start this presentation with an interesting schematic that shows the time course of evolution of the microbes who were the dominant life form in the oceans of the planet over billions of years, and only relatively recent in evolutionary times, 500 million years ago, the first animals, marine animals, the form of the hydra appeared. You may wonder why that is of interest to this presentation. Well, if you look closely at this animal, it is essentially a floating digestive tube with a mouth on one side and an anus on the other side. And it's wrapped in a nerve net that surrounds this animal and provides it with propulsion and activity that, as you'll see, is very close to what the enteric nervous system does today. There's a further step in evolution, which took another 500 million years before humans developed two brains, one located in the gut, the enteric nervous system, and one located in the brain with a division of labor. Just to illustrate the remarkable similarities in design, this is again the hydra in a petri dish, and you can see this peristaltic contraction that moves the contents through this tube from one end, the oral end, to the anal end. And you can see here the design of the nerve network in comparison to a display of the architecture of the nerve networks that make up our enteric nervous system, the nervous system of our gut. And in evolution, this was really our first brain. Our enteric nervous system is also our first brain. And in evolution, with the development of polar organisms, ultimately the human being, a second brain developed using many of the blueprints from neurotransmitters and connections and principles of communication from the enteric nervous system. This is again illustrated in a little bit more detail on the left side, cross-section through the gut with some of the enteric nervous systems, the enteric neurons shown here of the myenteric plexus, which regulates basically contractions, submucosal plexus, which regulates secretion, separated by the circular muscle layer. And you can see that there's sensory nerve endings that go into the villi of the gut epithelium. On the other hand, there's our brain, our main brain, with emotion regulating networks. And in the center of this are the gut bacteria that can produce substances that interact with cells in the gut, located, shown here, which contain neurotransmitters such as serotonin or other neuroendocrine substances and hormones. This interaction of the gut bacteria, which play a major role in the release and synthesis of serotonin, has an influence both on the gut side in terms of motility and secretion, and on the brain side in terms of emotion and many other vital functions. So we basically understood the system of brain and gut and the little brain of the gut fairly well and thought that we could explain most of the basic physiology of digestion, also of abdominal pain when there's a dysregulation. But then came on the horizon the microbiome science, which revolutionized many of our concepts about normal physiology. And without getting into the details of it, I just want to emphasize one point here. There's 40 trillion of these microorganisms in our gut, the greatest abundance on the planet in one location, in the large intestine. But most importantly, if we compare the information that's contained in our human body, in the genes of our human body, about 20,000 genes, with up to 20 million microbial genes, many of which we don't know the function of, we realize that we have neglected, but that we are still neglecting about 99% of that component of what's called a holobiont. So the combination of human and microbial genes, and only 1% has made it into our textbooks and our therapeutic approaches. This schematic shows you the red line are the human genes in representation of their numbers, and all the rest of this body would be made up by microbial genes. So what do these genes allow the microbes to do? I showed you earlier that microbes were the dominant life form in the oceans of the planet. And what they did over billions of years is they perfected their ability to communicate with each other, because there were only microbial organisms there, and later with animals, with marine animals like the hydra. When we first thought about the function of microbes, it was a very simple concept. So essentially, microbes would break down resistant starch or complex carbohydrates into short-chain fatty acids, which would then, regulate gut functions, have anti-inflammatory effects on the gut-based immune systems, but that also apply to other molecules like lignans, also xenobiotics, or chemicals that microbes have never seen in evolution, but that they are able to metabolize, most of them, given their large genetic variety. They can also break down proteins into amines through fermentation. Now, for a long time, it was thought that these processes are localized at the gut and affect gut function, play a role in gut dysfunction. In the meantime, we know that most of these molecules actually can have signaling functions to other organs in the body and other organ systems, such as the immune system, the cardiovascular system, the enteric nervous system, obviously being the closest, metabolism, and also with the brain, signaling to the brain and influencing behavior, which was first discovered about 10 years ago in spectacular animal experiments showing that the mouse behavior, emotional behavior, or emotion-like behavior could be influenced by the microbes. System has evolved to a great complexity. We know that these trillions of microbes with their millions of genes that they have can transform a variety of source materials, which I show you in a minute, into hundreds of thousands of metabolites, and that many of these metabolites are released into the bloodstream, where they're mixed with host-derived metabolites, some of which stay in the gut and act locally. But essentially, this vast and very complex communication system can influence every organ within our body. So here are the sources for these gut microbial signaling molecules. Many of which are diet-derived, so complex polysaccharides, complex carbohydrates, polyphenols, amino acids come with our diet, and they're transformed by microbial transformation into signals such as short-chain fatty acids, simple phenolic acids, estrogen metabolites, and various neurotransmitters. There's also host-derived sources, so like primary bile acids, steroid hormones, female and male sex hormones can all be metabolized after they're secreted into the into the ileum with the bile, they can all be transformed into signals like secondary bile acids and active hormone metabolites, which have been shown to circulate throughout the body and also play a role in as distant sites as the brain. Then there's microbe-derived components like cell wall components. So if microbes break down, or even when they're intact, these cell wall components are being transformed into signaling molecules, used as signaling molecules to the immune system, a major trigger for immune activation. And then there's these synovial molecules, xenobiotics, I mentioned as toxins, biosynthetic precursors that can also be transformed into toxins, or they can be detoxified. So a wide range of sources for microbial signals, which create a very complex communication system that we're just beginning really to understand. One example, practical example in terms of diet-derived sources, is an example of the traditional Mediterranean diet with a large proportion of fruits and vegetables, which are transformed by microbes, mainly in the large intestine and in the distal small intestine, into, as I showed you previously, into these metabolites that are basically then signaling molecules. So in plants, polyphenols, phytochemicals, and fiber being the main sources for these signals. So what we eat has a major influence on the signaling, on the type of signaling and the signaling capacity of the system. There's basically three transmission modes of microbial signals: neurocrine, which acts locally on afferent nerve terminals, either of extrinsic nerves, such as the vagus nerve, or on sensory nerve endings of IPENs, intrinsic primary afferents of the enteric nervous system. There's endocrine signaling, where molecules are released into the systemic circulation and acting distally. So satiety signals are a good example of that. As a major trigger, have, for example, short-chain fatty acids acting on receptors on neuroendocrine cells. And then there's these systemic immune mediators acting locally and distally in form of cytokines, membrane-associated structures, and molecules like LPS, lipopolysaccharide. They all can activate immune cells. Now, if you look at what these signaling molecules, once they get to the brain, so just picking one site in the body where the signaling molecules can act on as part of the brain-gut microbiome system. In the brain, schematically, we have neurons, we have glial cells, oligodendrocytes, astrocytes, microglia, and we have endothelial cells in the vasculature of the brain, also being part of the blood-brain barrier. And what I'd like to show here in this slide from a paper by Needham is there's beneficial effects of microbial signaling molecules, such as short-chain fatty acids, which generally have anti-inflammatory effect. And you can see that short-chain fatty acids come up in all these boxes, acting on these different cell types in the brain, having beneficial effects. And you can also see that, for example, secondary bile acids play an important role, most of them beneficial, some of them, in particular the ratios between certain bile acids, may even have negative effects on the brain. And then you can also see the polyphenols, these molecules that are also a major component of plants and fruit, which are broken down by the microbes into short phenolic acids and molecules. On the other hand, we have microbial metabolites that have detrimental effects, such as these membrane-associated microbial patterns, so molecules that are in the membrane of microbes, like LPS, lipopolysaccharide. We have simple phenols. And you can see, again, just like as we had this consistent effect of positive microbial metabolites that provide a benefit to these cells, we have a series of these molecules, particularly with ability to activate inflammatory systems in the brain that have detrimental effects. So you almost could say it's the balance between these positive and these negative influences that the cells in our brain are exposed to that determines the health of the brain. And you could extrapolate a similar model to other organs in the body, such as the heart, such as the lungs, and the liver. Now, let me give you one example of this neurocrine signaling pathway I mentioned earlier as one of the communication channels. So we have the brain connected to the gut. You remember this from an earlier slide. These enterochromaffin cells that contain tryptophan hydroxylase, the rate-limiting enzyme for serotonin synthesis from tryptophan, from dietary tryptophan. These cells have these extensions, these neuropods that form synaptic connections with vagal afferents, but also with efferents of the autonomic nervous system coming from the brain. Interestingly, the microbes play a major role in stimulating through activation of short-chain fatty acid receptors and secondary bile acid receptors, stimulate this enzyme. So the more of these microbes exert their influence, the greater the synthesis of serotonin is. This, again, is influenced by the presence of dietary tryptophan. And to make the whole system even more complex, there's not just release of serotonin on the basolateral side going to these vagal afferents, but serotonin is also released into the gut lumen and interacts with molecules similar to the serotonin reuptake inhibitor, which are localized on certain Clostridialis microbes that can take up the serotonin and incorporate this into the cell of the microbe. Microbes themselves don't produce serotonin, so they use this circuit basically to generate their own serotonin levels. There's another role for microbes in generating another tryptophan metabolite, kynurenine. It's again a process that starts with tryptophan. This conversion, this metabolism is modulated, is inhibited by hydrogen peroxide coming from lactobacilli. This hydrogen peroxide affects the rate-limiting enzyme of kynurenine synthesis, this IDO1 enzyme. So if there's a lack of hydrogen peroxide, for example, with a decrease in the prevalence of the lactobacilli during chronic stress models, there's an increased production of kynurenine. And finally, there's a third arm, the production of indoles from tryptophan. Now, this is different because indoles are not just modulated, the indole production is not just modulated by microbes, it's totally dependent because only microbes have the rate-limiting enzyme to produce indoles, a variety of indoles. One has attained some notoriety, indoxyl sulfate, which has been identified in playing a role in several diseases, brain diseases like autism spectrum and Alzheimer's disease. So brain-gut microbiome interactions is not a static system. It is definitely a system that evolves during development over a lifetime. And there's different phases that play a significant role. So early on in life, multiple influences that have been characterized. I won't have the time to get into this, from availability of breast milk, C-section born versus vaginal born, exposure to antibiotics early on in life, and the state of the pregnant mother in terms of maternal infection, health, and also metabolic status. And then there's, in addition, environmental influences, the stress that's exerted on the infant and on the pregnant mother, all of which have been shown to influence the programming in the first 3 years of the gut microbiome. It's assumed that these processes play a role in the pathophysiology of several developmental disorders, such as irritable bowel syndrome, anxiety disorders, depression, ingestive behavior, and autism spectrum disorders. One way to look at this is in a systems biological way that early influences shape the gut microbial ecosystem with stable states. For example, this stable state 1, which would be associated with a healthy, diverse, and rich microbiome, creating stability that even in response to perturbation, it would not change the main location of the system. And the factors that influence the development of this landscape, this ecosystem landscape, are diet, early life experiences, genetics, and antibiotics. Now, later in life, we have influences, particularly diet, medications, and influences from the brain, like stress and depression, that can modulate the existing initially programmed microbiome. And it's thought that these influences play an important role in metabolic disorders, and particularly in neurodegenerative disorders such as Parkinson's and Alzheimer's and early cognitive decline. So we come back to this, this ecological landscape, this stability landscape. In the adult, the factors that exert perturbations are diet, like the unhealthy standard American diet, influences from the brain, psychological stress, emotions, and lifestyle factors, including things like social connectedness and exercise, shown in a different way here. So from the early onset of life, where an influence of sex and race is exerted on the brain-gut microbiome system, multiple perinatal factors that play a role, early adverse life events, allostatic load psychologically and diet-wise during adulthood. And gut microbial metabolites are probably involved in some way at all these stages, as is evidenced by current data obtained both in animal models and in human studies. So ultimately, the microbes have an influence on brain development in connection, in concert with these other factors. And ultimately, this interaction results in the formation of brain circuits, or brain networks that are responsible for basic functions such as motivation, saliency, stress, and resilience, and others. Ultimately, this is the basis for brain disorders with altered brain networks. And what is currently a main focus in the research is what role do these gut microbial metabolites, either early in life, during adulthood, or late in life, have in shaping or disturbing the normal development and function of these brain networks. So let me show a few things of three examples of perturbation of the brain-gut microbiome system. Where we talked about stress, emotion. So we know that the autonomic nervous system responds to both psychosocial stress from outside, but also internally generated stress, negative emotions in a variety of ways. And these stress-induced changes change the microbial environment because they change the habitat where the microbes live in. So they change GI motility, regional alterations, intestinal fluid and mucus secretion, bile acid secretion, enteroendocrine function, and so forth. So these factors, this factor of allostatic load or psychological stress plays a major role, indirect role, on the composition, function, relative abundances of the gut microbiome. And there's many studies both in humans and in animals that have demonstrated this. In addition, there are direct stress-induced effects on microbiota function shown here. So norepinephrine has been shown to act on special receptors on gut microbes, quorum sensing receptors, which stimulate gene expression in these microbes and alter their behavior towards the host. So it's been demonstrated for noradrenergic signaling, but Also, there's indirect evidence for stress-induced signaling using serotonin, opioids, and probably other molecules stored in these enteroendocrine cells, enterochromaffin cells in the gut. What role does the gut microbiome play in mediating the influence of chronic stress on the immune system? Well, if you look at a cross-section through the gut, so we see the epithelial layer makes up the gut wall with some enteroendocrine cells. We have a thick mucus layer of two separate layers, different functions. And we have the immune system just microns away from the inside of the gut, just separated by this mucus layer. And then we have in a healthy gut a highly diverse and rich ecosystem. So this intact gut barrier separates the sensors of dendritic cells from coming in contact with the membranes or these MAMPs of gut microbes. Now, what happens with chronic dietary and psychological stress? So the impact is actually kind of similar. So we have a thinning of the mucus layer. Microbes now come in contact with toll-like receptors on dendritic cells, which stick into the mucus layer. This sends signals to the gut-associated immune system and activates both the adaptive and the elements of the immune system. Cytokines can act back on the barrier that's created by these tight junctions, loosening these tight junctions, allowing intact microbes penetrate or translocate through the gut epithelium and coming then in direct contact with a variety of immune cells located in the gut. So both the innate and the adaptive immune systems are involved or are now engaged. The result of this is an increase on the one side in molecules generated by immune cells, but also in the translocation of intact microbes or microbe membrane components into the systemic circulation, creating a state of what's called metabolic endotoxemia without an actual infection. And in the presence of an increased genetic risk or other risk factors, it plays a major risk factor in the development of a variety of diseases that are seemingly unrelated, but which we're beginning to understand have certain common pathological factors that play a big role in them. Showing this again, what happened with the changes that have occurred with our changed lifestyle in the last 75 to 100 years, in terms of diet, stress, hospital, the way we come into this world and are exposed to antibiotics in hospital and various medical practices, creates a state of dysbiosis, dysregulation of the microbial ecosystem. And that is a process that happens fairly quickly because microbes can adapt within days in their relative abundances, diversity, and function to changing influences due to their large number of genes. So the microbes rapidly adapt, but our gut and its immune system is much, much slower. Takes 10 to 15,000 years till we really have genetic changes in our human, in the host cells. And this is now perceived by the immune system of the host as a mismatch, as a threat to the homeostasis. This altered gut microbiome is no longer something that these immune cells recognize or are familiar with, and they respond as if this were an infection. And the outcome is that, as I said in the previous slide, a variety of inflammatory molecules, both membrane components, but also various cytokines get into the systemic circulation and can affect many organ systems. Summarized schematically here, so we can look at the brain-gut microbiome system. I prefer the expression system because these nodes of the system are connected in a bidirectional way with feedback loops. So nothing in the system responds in a linear fashion. And this system is exposed to both brain-targeted stressors, the psychosocial and emotional stressors, and dietary stressors, which are basically the dramatic changes that have occurred in our in our food system and in our dietary habits. And ultimately, both of these influences result in the alarm bells go off in our immune system, in the gut-associated immune system. The majority of immune cells travel or are located in the gut and travel from there to other organs. So this system is probably the most important system in our body that transmits environmental factors, perturbations into internal changes and which play a major role in disease pathophysiology. So the combined allostatic load or persistent stress-related stimulation exert a detrimental effect. So acute stress is something that has very positive effects, has saved life on the planet Earth for millions of years. But if it becomes chronic and if it comes in a combined way, as we experience in this today, what's been observed is increasing allostatic load. Combined allostatic load is associated with a variety of changes in our body, in particular this chronic systemic immune activation. And this systemic chronic immune activation seems to be playing an important role. It's not the cause, but it plays an important role in most, probably all of the modern chronic non-transmittable diseases, the epidemic that we have been experiencing increasingly during the last 50 to 75 years, which we have responded to with massive interventions in terms of pharmaceuticals and the medical system and surgeries. So the human mortality has generally not decreased, maybe with the exception of the recent couple of years with the COVID-19 and just drug-related deaths. But in general, the longevity has increased for people. However, it's not a healthy longevity, but it's longevity that is bought essentially with billions of investments in medications and chronic medical care. Not just because I'm a gastroenterologist, I like to emphasize so that this concept of gut health, the way I explained this to you, the interaction of our diet, the microbes, and the immune system, and the effect on the rest of the body has been shown to play a role in all of these diseases. And the immune system, even though there's other connections, as I showed you earlier, in terms of the neurocrine and the endocrine, it appears that the immune signals play the most important role in contributing to these chronic diseases. So let me give you a few examples, and I'll go through this quickly. Mood and affect. The earliest studies with in mouse models have shown that the microbes play a role can play a role in the regulation, modulation of emotional behaviors, summarized here. So earliest findings were that if you do a fecal microbial transplant from a particular mouse strain that is, you can call timid or introvert, into a germ-free animal that has a different background in terms of emotional reactivity, then this animal that receives the fecal transplant will adopt the same type of behavioral pattern as the donor mouse. This can be reversed with another microbial transplant from this mouse. So this has not just been done as the early experiments in mice, but it's also been shown that you can do a transplant from humans into mice, creating the same depression-like behavior. You can identify individuals simply based on the composition and relative abundances of their fecal microbiome in terms of who falls into the category of major depressive disorder and which stool comes from individuals that are healthy. These are the relative abundances of these microbes. And in the mouse, interestingly, this same pattern is preserved. So those mice that got a transplant from an individual with depression also showed the depression-like behavior as opposed to a mouse that got the transplant from a healthy donor. And this has also been followed further in terms of metabolites that differentiate these two populations depending on from which human population the fecal microbial transplant came. There's several studies like that, often with contradicting results. So we've not really accomplished to identify a global general signature of depression at the gut level, even though larger studies have addressed this and have revealed it's the subsets of patients with depression that have different gut microbial signatures. This was a very large study where the investigators tried to address this very question. Is there a neuroactive signature, or is there a gut microbial signature that characterizes quality of life and depression? The study came out negative, very large study, 1,000 individuals. It did not show a depression-specific signature, but the gut microbial composition, relative abundances, and function were associated with quality of life measures. So it did not quite prove what the investigators started out with, but it did show a relationship between microbial ecosystem composition and quality of life measures. So what we think today that is likely a scenario that in individuals with a genetic predisposition that are exposed to different kinds of stresses during life, starting with early adverse life events, trauma, chronic stress, this will lead to a change in what I like to call the gut connectome. So combination of epithelial cells, neuroendocrine cells, and the enteric nervous system and immune cells, that this influence of the autonomic nervous system and the HPA axis will change the function of this gut connectome in the form of changing the composition, relative abundances, increasing the epithelial permeability, as we talked about earlier, in terms of this leaky gut, and in terms of immune activation. These gut changes lead to the generation of microbial neuroactive metabolites and immune mediators that feed back to the brain. And we don't know exactly what role this feedback plays. Does it play a role in specific aspects of depression, or does it play a role just in the chronicity of depression and the difficulty of bringing somebody from a state of depression back to normal? So a few words about obesity and digestive behavior. Several intriguing mouse studies with antibiotics, probiotics, fecal microbial transplants, germ-free animals. There's now some reverse translational studies from transplant of fecal material from humans to mice. And there's studies, human bariatric surgery studies, all of which support the concept that altered brain-gut microbiome interactions play a role in some form of obesity, in particularly in an entity called food addiction, which is fairly common in patients with increasing BMI and body weight. The same sort of circular model or systems model that I show for depression, that in individuals that are genetically or epigenetically predisposed in their hedonic system and their appetite regulation systems, if they are exposed to high fat, high sugar, high caloric foods, as we see it in our society, this will lead again to a change in the gut microbes, in the increased permeability, this leakiness of the gut, this generation of local inflammation and general inflammation, and also to neuroplasticity of some of the sensors of extrinsic nerves that innervate the gut, so they become less responsive to some of the satiety hormones that come from enteroendocrine cells like cholecystokinin. Then these diet-induced alterations in gut-brain signaling, similar to what I said for depression, generate a variety of signals, a reduction of satiety signals, which then feed back to the brain. We know, for example, that the satiety mechanism in the hypothalamus are compromised by activation of immune cells, glial cells in the hypothalamus. So a similar mechanism as I showed you earlier for the brain. The result is food addiction and craving for high caloric food. We don't know to what degree, to what percentage these microbial components play a role in these dysregulations. If it's mainly by creating the chronicity of the problem and the difficulty of reversing it, or if it creates specific behavioral features in an individual, like this craving for high caloric food. Parkinson's disease, another one of the diseases where the gut microbiome, the brain gut microbiome system has been implicated. This has been recognized early on by Mr. Parkinson, who already felt that a disordered state of the stomach and bowels may induce a morbid action in a part of the medulla spinalis. And what we know today is that most patients with Parkinson's disease are constipated. We also know from epidemiological studies that the constipation is a very early sign, can predate the onset of neurological symptoms by 15 years or more, that these early non-neurological symptoms predate the full-blown syndrome for a long time. We also know that these individuals at this stage have altered gut microbial composition and features, both in terms of their relative abundances as well as the metabolites that they produce. And another intriguing finding is that the first degenerative signs in a large number of patients with Parkinson's disease occur in the enteric nervous system neurons. This alpha-synuclein aggregation occurs in these myenteric cells, which interact with the vagus nerve, and this alpha-synuclein is transported through the afferent branch of the vagus nerve, initially to the brainstem areas, where the locus coeruleus, for example, is one of the first sites that is compromised, leading to this REM sleep abnormality, but then later migrating up into higher centers, such as the basal ganglia and cortical and emotional networks within the brain. So we also know that individuals that underwent a truncal vagotomy had a lower incidence over time of developing Parkinson's disease, shown here. And this was only seen with truncal vagotomy and not with highly selective vagotomy, where this innervation of the gut would be left intact. So very strong evidence, I would say, of all the diseases where an alteration of the brain-gut microbiome system has been implicated, very strong evidence for a major role in the development of Parkinson's disease. And the good news is that is this long window where an intervention might be possible to prevent or slow the development of the neurological disease once we understand more the mechanisms that are involved. So the last example, and I won't get into the detail of that, there's also through the work of Elaine Chao and Sarkis Mazmanian, this identification of an altered gut microbiome in mice with autism-like behavior. This has been pursued in startup company and some early experiments, largely uncontrolled, that have shown that human fecal microbial transplantation can actually reverse some of the symptoms, both the gut symptoms that patients with autism spectrum have, as well as the neurological symptoms. So there's many challenges that remain. Summarize some of them here. So the mouse brain is obviously very different from the human brain. The whole role of the prefrontal cortex that mouse has very underdeveloped is not taken into account when we extrapolate from mouse experiments. The mouse behavior, simple behavior versus complex human emotions, which in some ways we make this comparison in our translational models, but there's really no definite proof that there is a close correlation between both the mechanisms and the outcomes of interventions. It's another one, human clinical variability versus inbred mouse strains raised under identical conditions, which creates this big challenge of either having to study a very large number of human patients or selecting, highly selecting subtypes of human pathology in which to perform the studies, which has not really happened to a significant degree. One approach that has been tried is this big data approach, identifying patient subsets with then providing a basis for targeted, personalized interventions. There's also still a major issue with the fact that most studies to date in humans are cross-sectional associations and much fewer are longitudinal interventions. Very few studies are with interventions that would allow a conclusion about causality between microbial composition and function and human disease. And then there's also one, I mentioned this a few times, that a systems biological approach is necessary to understand the complexity of the gut microbiome system. We're still relying in most of our experiments and conceptualization on linear or one-directional influences, not taking into account this principle of circular systems with multiple reflex loops. And finally, gut microbiome assessment. There's been tremendous progress in this area with development of a variety of functional assessments from metabolomics, metatranscriptomics, which all have, in some ways, are replacing the earlier studies with 16S sequencing that have just looked at the relative abundances of microbial species. And there's also this development of new bioinformatics approaches with artificial intelligence, neural networks, to understand the complexity of the gut microbiome. I hope I've convinced you that the simple model that early on was picked up by commercial entities, but also by some scientists, that there's a simple relationship between individual microbial species and human pathology, that this is definitely not the way that we will make major progress or have major therapeutic inroads in this area. So one could ask the question, is the microbiome causing a fundamental transformation of brain science? I would carefully say yes. The answer is yes. So this fundamental transformation, looking at this sentence by Thomas Kuhn, science is a series of peaceful interludes punctuated by intellectually violent revolutions, in each of which one conceptual worldview is replaced by another. It could well be, when we look back in a few years, that this microbiome science has caused such a violent revolution in our understanding. So if you're interested after this whirlwind tour to learn more about the concepts that I laid out to you, so you're welcome to get one or both of my books, which are sort of complementary, or you can just go to our website and sign up for the biweekly Mind-Gut Connection newsletter. Thank you so much for your attention.